What if artificial intelligence could discover entirely new ways to fight cancer that human scientists never thought of? Well, that just happened. Google Deepmind and Yale University announced something remarkable recently. An AI model that didn't just analyze existing cancer research, it generated a completely new hypothesis about how to treat cancer. And when scientists tested it in living cells, it actually worked. Reading the language of cells. The Google and Yale team began with this approach. What if an AI could read cells the way ChatGPT reads words? Not just detect patterns, but understand what cells are saying through their genes. Every cell in your body runs on a code, thousands of genes turning on or off in specific patterns. Scientists can measure this using single-cell sequencing, but the result looks like a spreadsheet with tens of thousands of numbers per cell. It's an unreadable mess to the human brain. So the Google-Yale team thought, what if we turned those gene activities into sentences and trained an AI to read biology as if it were text? So the team built an AI model called C2S scale, short for cell-to-sentence scale, to do just that. Each cell's gene expression pattern becomes a sentence made of gene names, and the AI learns the grammar of biology. Just like a language model learns that the cat sat on the mat makes sense, C2S scale learns that interferon up, antigen pathway active, means the immune system is engaged. They trained the model on millions of single cells combined with biological and experimental data. As the model grew, up to 27 billion parameters, something remarkable happened. It started reasoning about cellular behavior. It could predict how a cell might react to a drug or immune signal, even if it had never seen that exact experiment before. In other words, the AI wasn't just memorizing, it was actually learning biology. The weak signal problem. Let's examine the problem this AI was trying to solve. Imagine your immune system as a highly trained security force patrolling your body, looking for threats. Normally, when a cell turns cancerous, it displays warning signs on its surface, called antigens, kind of like alarm bells. Your immune cells spot these warning signs and eliminate the threat. Now, there's a spectrum of how tumors interact with the immune system. On one end, you have cold tumors. These have essentially no immune cells around them, no immune activity at all. They're in complete stealth mode, hiding from the immune cells. But there's another challenge that's perhaps even more frustrating. Warm tumors, where your immune system has shown up. Immune cells have infiltrated the tumor area. There are immune signaling proteins like interferon present. But the signal is just too weak. It's like your alarm system detected something and dispatched security, but the alarm is so quiet that nobody can quite tell where the threat is or how serious it is. This is a huge problem for immunotherapy. Even when the immune system is partially activated, if the cancer cells don't display enough antigens on their surface, the immune response fizzles out. The immune cells are right there but can't get a clear enough signal to mount an effective attack. Scientists have been trying to figure out how to amplify these weak but present immune signals, how to turn the volume up on that quiet alarm so the warm tumors can be detected as hot tumors, and that's exactly where this AI comes in. See the challenge and dual context screening. Here is the specific challenge the researchers gave to C2S scale. Find a drug that acts as a conditional amplifier, one that boosts the immune signal only when there's already some immune activity present, specifically when there are low levels of interferon, but not enough to trigger effective antigen presentation on its own. This is incredibly tricky because the drug needs to be context-dependent. It should amplify weak immune signals, but not act when there's no immunity activity at all. That distinction is crucial for cancer therapies because you don't want a drug that blindly activates immune pathways in every cell that could cause dangerous autoimmune side effects. Let me give you an analogy for how they tested this. Imagine you're trying to find the perfect sound system amplifier for a restaurant. Context A, the busy restaurant, with quiet conversation. You have a restaurant with some customers having conversations, but everyone's speaking so softly that the atmosphere feels dead. There is sound, but it's too quiet to create a lively ambience. You want an amplifier that can pick up those existing conversations and boost them just enough to create energy in the room. Context B, the empty restaurant. Same restaurant, but it's completely empty, dead silent. No customers, no conversations, nothing. The challenge is to find an amplifier that makes the quiet conversations in the first scenario more vibrant and audible, but does nothing in the empty restaurant, because amplifying silence just gives you louder silence. You can't amplify what isn't there. This is exactly what dual context screening does. The AI analyzed over 4,000 drugs in the two different scenarios. Immune context, positive or context A, real patient tumor samples where cancer cells and immune cells were already interacting, with low levels of interferon present, an immune signaling protein, but insufficient to trigger strong antigen presentation. Immune context, neutral or context. B, isolated cancer cells in a lab dish with no immune system components present at all. The AI had to find drugs that would amplify the weak immune response in the first context, but do nothing in the second. This required genuine reasoning about cause and effect, understanding that the drug only works when there's already an immune foundation to build on. So what did it find? Among the top candidates was a drug called Silmitasertib, also known as CX-4945. Now, this drug was already known to scientists. It's a kinase inhibitor that had been studied for cancer. But nobody had made this particular connection before, that combining it with low-dose interferon could dramatically boost how cancer cells display antigens to the immune system, but only when there's already some immune activity present. The AI predicted that Silmitasertib would amplify immune signaling by about 50% when combined with low-dose interferon in the immune context positive setting, but would have little to no effect when applied alone or in the immune neutral context. In other words, the AI predicted Silmitasertib alone, no major change. Interferon alone, slight increase in immune visibility. Both together, about a 50% boost in antigen presentation, the key process that lets the immune system see the tumor. Here's the kicker. Yale scientists tested this prediction in actual human cells, cells that were never part of the AI's training data. And it worked. When they treated cells with Silmitasertib alone, nothing happened. Low-dose interferon alone had only a modest effect, but combining the two increased antigen presentation by nearly 50%, effectively amplifying that weak immune signal into something the immune system could actually act on. This is crucial because it means we might be able to help patients whose immune systems have already infiltrated their tumors, but aren't mounting a strong enough response. It's not about creating immune activity from scratch. It's about taking the activity that's already there and making it more effective. D. Why context? B still matters. You might wonder, why include context B during the screening, the version with no immune activity? Because it's the control group, the baseline that proves the AI isn't being fooled. If a drug works in both contexts, it's just a blunt tool that works indiscriminately. But if it works only in context A, then it's a conditional amplifier, the kind of precision behavior we want. It's like testing a noise-cancelling microphone in both silence and noise. You need both tests to know it's truly adaptive, not just always on. E. From cold tumors to hot tumors, the two-step strategy. Now, here's the key insight. The dual context screen doesn't create immune activity, instead it builds on it. To turn a cold tumor hot, you need two steps. Step 1. Priming. Spark the immune system using something like a vaccine, checkpoint inhibitor or low-dose interferon. This could turn the cold tumor into warm status. Step 2. Amplifying. Once that spark exists, use a conditional amplifier, like Silmitasertib, to fan the flames. This could now turn the warm tumor into hot status. C2S scale helps scientists find those Step 2 drugs that cooperate with priming the immunity response safely and effectively. Why this matters. Here are the major takeaways from this new discovery. First, it's not just finding patterns in existing research, it's generating genuinely new hypotheses. The AI reasoned through complex biological conditions and came up with an idea that human researchers hadn't thought of before. That's different from just being a really good search engine. It can come up with novel ideas. Second, the new hypothesis was tested and validated in living cells. The discovery wasn't just theory, it's a real biological mechanism that works in the lab and could potentially lead to new cancer therapies. Third, it points to a specific clinical application, helping patients whose tumors already have some immune infiltration but aren't responding well enough to immunotherapy alone. This could be particularly valuable for combination therapy strategies, where you might first use treatments to get immune cells into the tumor environment, then use this amplification approach to make those immune cells more effective. Fourth, it demonstrates a blueprint for a new kind of scientific discovery. We can now use AI to run virtual drug screens on thousands of compounds, generate hypotheses about how they might work in specific biological contexts, and then test and validate the promising ones in the lab. This could dramatically accelerate drug discovery. Yale researchers are already exploring the mechanism further and testing other AI-generated predictions. If these findings hold up through additional preclinical and clinical trials, we could see new combination therapies that amplify weak immune responses, helping the immune system battle cancer cells more effectively. We're witnessing AI move from being a tool that helps us find needles in haystacks to being a partner that helps us imagine entirely new needles we should be looking for. And when it comes to fighting cancer, this could make substantial progress in our battle. If you found this helpful, give it a like and subscribe for more science and AI wonders. Until next time, thanks for watching.